Hi, so we're going to go through each of our steps for our muscle contractions. It's going to be a really great idea for you to write all this down in your notes so you're able to have this information when it comes to our test. So the first thing that we're going to start with is step 1. So we're starting at the initial part of this muscle contraction, and that initial part is going to be that signal that arrives in the neuromuscular junction. So if we break down that word right there, neuro and muscular. So we know that muscular obviously is going to stand for muscle, but neuro here is going to stand for our nerves. So we need to get a signal from our nerves to tell our muscle what to do. And how we're going to do that is through something called neurotransmitters. So these neurotransmitters are going to move towards the motor end plate. And if we remember from our notes, our motor end plate is a part of that nerve that is going to connect to our muscle fibers itself. So then once those neurotransmitters move to that motor end plate, the neurotransmitters which is going to be called acetylcholine. Let me just spell that right. Acetylcholine. That's going to be our neurotransmitter here. That is going to be released into the synaptic cleft. And the synaptic cleft, again, is just that little space between our neurotransmitter, or the little gap between our nerve cell, our motor end plate, and our sarcolemma, which our sarcolemma, if we recall, is the membrane that we're going to see around our muscle fibers. So let's draw a little picture here. So let's make this our nerve. I'll keep it consistent with the blue and red here. And this is going to be our sarcolemma. So right here, this is our sarcolemma. And this is our motor end plate. Okay, so what's happening here is that we're going to see little vesicles inside our motor end plate that are going to be carrying our neurotransmitters. And once these are going to diffuse across the membrane, we're going to see that our neurotransmitters are going to be all around here. And we're going to see little receptors that are going to accept those neurotransmitters. So here's our step 2 here. Step 2, acetylcholine binds to receptors on the sarcolemma. So let's add to our picture here. This rainbow color is going to be our receptors. And we're going to see that this acetylcholine is going to come and bind here. Okay, so this is a good picture when we're thinking about the first 2 steps of our muscle contraction. But if we keep going here, we know that our sarcolemma is that membrane of our muscle tissue. So step 3 is we're going to be looking at some other things that are surrounded those muscle fibers, which is our sarcoplasmic reticulum and our T tubules. So what we're going to see next happen is an action potential is sent through that sarcoplasmic reticulum and T tubules. So remember, our sarcoplasmic reticulum is going to control the amount of calcium that enters our muscle fibers. I'm going to abbreviate calcium here, C+. So it controls the amount of calcium in muscle fibers. But it's not going to release this calcium until we see this action potential happen here, which is going to bring us to step 4. So for step 4, we're going to see that calcium, C+, is released. So this releasing of calcium is going to do two things here. So the calcium is going to bind to troponin. If we remember, troponin is a protein that is wrapped around our actin. And when it binds to this troponin, the troponin is going to change shape. That's going to cause something called tropomyosin, which is covering binding sites on actin. It's going to move off of these myosin binding sites on the actin. Okay, so calcium is now released into the muscle fiber. That calcium is going to bind to troponin, and that troponin is going to change shape, which is going to cause the tropomyosin here to move off of our actin binding sites. So our next step here, so I should say actin. So our next step here, step 5, is going to be that the myosin heads that are charged with ATP, which is our energy molecule here. ATP is super important. So those myosin heads charged with ATP are going to bind to the binding sites on our actin. And perform a power stroke, which is also going to be a really important term here. So let's take a look. Let's draw out a nice picture here for our power stroke. So here I'm going to have our myosin. It's going to be nice and green. Remember, our myosin is nice and thick, so I'm going to make a bunch of lines here. And we're also going to draw in these heads that we would see. So here are our myosin heads. Green is myosin. Here we're going to make our actin molecules. And then we're also going to see our tropomyosin and our troponin around our actin molecules. Okay, so here's actin, and then troponin, and tropomyosin. Oops, sorry, those are supposed to be blue. Let's fix that up real quick. So troponin and tropomyosin. So here comes calcium coming in, and it's going to bind to our proteins here. And they're going to reveal some binding sites that we're going to see on our actin. So these are going to be displayed in these nice rainbow colors here. Here are our binding sites that are going to appear. So we have all those binding sites, and we're going to see that ATP is going to come in and bind to our myosin heads. Our myosin heads are going to connect to our binding sites, and these binding sites are going to perform that power stroke, pulling those two actin molecules closer together, decreasing the length between them, which is ultimately going to contract the muscle. When muscles are contracted, they are shorter. So contracted muscles are shorter. So that's because of that actin moving towards each other, decreasing the length of the sarcomere, which we know that our sarcomere is the measurement between two Z lines. So then our final step here, our step 6, is going to be when calcium is present. This attachment, so the power stroke, will continue until the muscle is fully contracted. So that's our last step here, is just that repetition of this power stroke. Myosin is going to detach from our binding sites, move to a binding site that is going to be further down on the actin, and pull it again closer and closer together until that muscle is fully contracted. This is a very complex process, so I really want to encourage you to write down these notes, draw out these pictures, watch that Edpuzzle video that I've sent to you, and really get all the information you need, and then come to me with questions on these, because this is all going to be a process that we're going to see with our test here. Okay, if you have questions, reach out. I will be here to answer those for you.